Good morning. Still morning. Yes, good morning. I'm Aline. I'm a conservation scientist here at the Botanics, and Erin and I will talk about some of the work we've been doing on the Whorled Solomon seal. And for a bit of an overview, this work we're presenting is part of a 3-year project supported by Scottish Government's Nature Restoration Fund. And all these species that you see on the pictures here are the species we're currently working on. They're all threatened for various different reasons, mainly habitat loss, also disease and hybridization. What skill set are we bringing to tackle the challenges we're facing to work with these species is conservation science and research. It's our basic principle. Then the expertise we have in conservation, horticulture and biosecurity, but also really important, the communication and partnerships across the project. And here we go. The steps we're taking are to first of all, understand what the problems are, do our research, and if required, collect material from the wild, bring it back to the nursery, bring it back to the nursery, bring it back to the nursery to bulk up the plants. That's Erin's job. Then we find suitable habitat. Lots of people in this room have actually helped us find habitat for the Whorled Solomon seal, for example, and then plant the species back into the wild. That sounds simple. It really isn't, as we've discovered. But all this work is also happening following best practice guidance. For example, the Scottish Code for Conservation Translocations that we follow. We're also working really closely with Nature Scot. Ian is there, our main project partner, on this project. And just to make sure that we're doing all the licenses, consents are in place, obviously landowner consent as well, before we do any work. Long-term monitoring, here we go. Long-term monitoring then helps us to refine our conservation strategies further. And finally, if we want to achieve anything at scale, Sarah mentioned it, collaborations and partnerships are really important. There's only so much we can do by ourselves. We don't own land, for example, at RBGE. So together we can do so much more. Right, so the Whorled Solomon seal. Here we go, Matt. You've seen it. In the UK, we have 8 or so populations that remain. They're all in Scotland. I'm not sure what the 2 dots in England are doing. That's quite right. But anyway, I think there's only Scottish populations. So quite rare. Consequently, its class is vulnerable. And while there isn't much information on historic distribution, it seems that the main issue is habitat loss. It's a woodland species. Also, if we compare that with its distribution in Norway, you can see they have a really good, a bit like the BSBI database, really, really good database on their species distribution. So that's Norway. And then you've got Scotland next to that. But also, some areas in Norway are really similar climatically to Scotland. So it can't be the climate itself. If we look at the GBIF at the European distribution, it's a very similar picture, quite widespread in other countries, really rare in Scotland. So we're trying to figure out what the problem is in Scotland, what's happening, and how we can help the species. And to do that, so that's the woodland site. And as you can see, so this is the site in Scotland. They grow in Scotland, it's just on steep woodland gorges or on the edges. And as you can see, they're quite prone to landslides. So the species likes free draining, nutrient-rich soils. And that picture is right next to a natural site, just missed the natural site, but they are quite prone to landslides. If we look at, again, populations in Switzerland, just 2 examples of woodlands I've seen there. The top picture, the ground cover, I don't know whether you can see, it's literally covered in Polygonatum verticilletum. And the bottom picture, again, across this woodland, which is more spruce forest, the ground is just sprinkled with Polygonatum verticilletum, but also frustratingly and interestingly, growing right next to Cucurbita alpina and Melamperum sylvaticum within the same square meter. Why? Anyway, and this is one example here from Norway, a population in Norway. growing right next to a path in the open, happily competing with shrubs, epilobium, grasses. So it shouldn't struggle that much, I think, in Scotland. Right, so to see what we can do for the species, we have three major aims. The first project aims to safeguard the species' large genetically diverse collections. And the fabulous Hort team, Erin, have done that, and they've propagated over 1,000 plants, including Scottish and foreign provenances. And Erin will tell you more about some of the complications that we can have in the nursery. And we have done some genetic analysis to inform the restoration work. Just one slide of on the genetics, I promise. Be glad to hear. But I thought it's quite interesting to share these preliminary results. So the analysis is still ongoing, but I wanted to share this. So I've looked at 15 microsatellite markers, so 15 locations on the DNA, and compared the plants with each other with these 15 locations. And if we just look at genetic diversity, and here shown as observed and expected heterozygosity, you can see that our two Scottish populations have a much lower diversity. Nil is low, and if you go, the higher the number, the higher the diversity. So our Scottish populations have a much lower diversity compared to Swiss and Italian populations, and also much lower than the Norwegian populations. But also, while I have analyzed 12 and 5 individuals, plants, from the Scottish populations, we just have one genetic individual in each population, which is not great for diversity and generally for population health. So diversity is really low in Scottish populations. So could it be that they've just gone through a massive bottleneck, which is why they're struggling in Scotland? One question. So our second project aim is to translocate the species back into suitable sites where they create resilient populations. And we have just completed that work literally 2 days ago and have planted 540 plants into 5 sites, which you can see on the map. These sites are both within and out with the species' natural range in Scotland to test for different habitats and climates. And following the genetic results, we decided to include foreign material as well to maximize the genetic diversity in sites and to see whether that makes a difference in terms of survival and vigor. And each site contains the exact same genetic composition, which means that any difference we see between sites is down to the habitat rather than the genetics. And all sites are also well away from natural sites because we're still trialing things. And so each plant is GPS tagged, and we have this fancy high precision GPS device that measures up to 30 centimeters. So we have quite precise data. And we use an easy free to use app, which is called Survey 123 for monitoring. And that is all streamlined. So even if we have different people monitoring in the future, we should have quite robust continuous data. And we end up also with really good shareable data. Everything is shareable, but also we end up with maps, which is really useful for if anyone wants to know what we've done and where we've done it, including ourselves. And this is a map here with one species, an example if we have 2 species next to each other. So they can be really useful. Each plant is individually tagged, so there should be no confusion with any natural regeneration or establishment. We have tiny tag data loggers that measure humidity and temperature, which we're also going to deploy in natural sites. So we have a comparison there in terms of the weather. And then we have different life stages as well that we've planted. We have seeds with flesh and without flesh. We have 1-year-old seedlings, and we've got established bigger rootstocks. And frustratingly, this is where it ends, because we have to wait for the first monitoring data to be able to say whether or not anything interesting has come out of this. But I'll be back. Also, if you wanted to keep in touch, our final aim is to inspire and engage people through stories of threatened plant recovery. We're trying to do that today, but also we've got Botanic Stories blog post series. So if you wanted updates on the project or see what else we're up to, you can do that. There's a QR code here that leads you straight to these stories. And you can also follow us on Instagram at RBGE Scottish Plants. And with that, I'm going to hand over to Erin, who's going to talk about interesting stuff.
Okay. Let's use this instead. So hi, I'm Erin. I'm one of the conservation horticulturists on the team, and I'm going to follow on from Aline and talk a bit about the horticultural aspects of conserving Whorled Solomon seal. So Polygonatum verticillum is a herbaceous perennial. It grows from a large rhizome. It can utilise different life strategies, so it can reproduce sexually or vegetatively. It's a woodland plant, so naturally grows in quite shady acidic soils, and it produces cream whorled flowers from May to July, which then develop into the dark red berries which contain its seed. We've cultivated over 1,000 plants in the nursery from direct sowing the seed and dividing rhizome collected from wild populations. Whorled Solomon seal has been one of the species we've found quite challenging to cultivate in the project. In the summer of 2023, all our collection of 1,000 plants came up green, stayed green for about 2 weeks, and then completely yellowed and died back. This worried the team quite a bit. We didn't know why this was happening. So we waited until summer of 2024 to see if it would happen again. And yes, it did. They came up green, stayed green for about 4 weeks this time, and then completely died back. And this was far earlier than they naturally should. So initially, the team suspected that this was some kind of plant virus. This is a major setback to the project. If we even suspected that our plants could have any type of virus, we absolutely could not plant it out, and our entire collection may need to be destroyed. Next slide. Biosecurity is really important to us, and we could not risk introducing a virus into the wild, especially while the future of the species is already so fragile. So in order to diagnose the problem, our team sent samples off to SASA, but they then declared it clear of a multitude of viruses. So with no real evidence to go off that this was viral, we decided to investigate if this was a cultural issue. And great news for us, because there's nothing more our team loves than a trial. So our lead horticulturist, Becca Drew, set up an experiment in which our 4 provenances in our collection were split equally between 12 trial groups. The aim of the trial was to see if anything cultural could reduce the yellowing rate, and through that, identify the ideal growing conditions for Polygonatum. So each of the trial groups had a different cultural variable controlled. So we controlled things such as shade, nutrient addition, media composition, wind exposure, and some of them were planted outside of their pots into the gardens. We also had one trial group, which was inoculated with mycorrhizal fungi, and this was taken from one of the natural Polygonatum populations. Our control group was just Polygonatum in our standard woodland mix, sitting out in one of the polytunnels, which is around 25% shade. You might be thinking, why not just replicate the conditions that the natural Polygonatum populations are found in? It's important to assume, not to assume, that this is what these plants like best. These are already quite fragmented populations, and it might just be that these are persistent at the edge of their niche. So for the trial, we collected data such as number of shoots, and we ranked each pot in a yellowing category from none to severe. And then we ran this all through statistical software to see if these cultural variables were having any real effect on our Polygonatum. I'd like to preface all this by saying our trial is not statistically sound, our sample size was not big enough, and our tests were not standardized, but it gave us a really good insight into how these variables were affecting our plants. And I'm going to hurry up because I've only got 5 minutes. So this is one of the findings here. This is all of our yellowing rates which were recorded. You can see that our media number 2 had the lowest yellowing rate overall. This was our most open and free draining media, which also contained leaf mold, so it's quite similar to how they'd be found in the wild. You can also see that our highest degrees of yellowing, so the darker shade of yellow there, was attributed to groups which did not have extra shade. So overall, this gives an indication that the ideal conditions for Polygonatum are free draining, open and shaded soils. Another finding from the trial was that we found that Polygonatum verticillum in association with mycorrhizal fungi produced on average the most shoots per pot. It might just be that the mycorrhiza is improving nutrient uptake and stress tolerance in these plants. A lot more research would need to be done into this, but if this were true, it could be used in future cultivation efforts to improve plant vigor. And the most interesting finding by far was that our cultural variables had actually no effect whatsoever on our seeding rate. However, a significant association was found between seeding rate and provenance. This may be that seeding rate is a result purely of genetics and not environment. In fact, it was our largest provenance on the right there that seeded the least. This is quite a concerning thought. If we'd had only the rye chip material on the right, we would have really struggled to build a seed bank in the nursery, and conservation of the species would have become a lot more complex. I hope this highlights to you the ways in which genetic diversity is absolutely crucial to conserving these Scottish endangered species. So what's the need to do trials at all? With many of the rare and endangered Scottish species we take care of, there's little to no cultivation protocols, and we're constantly learning the horticultural requirements of these plants. Knowing the ideal growing conditions allows us to select translocation areas with a higher rate of success. The nature of this project requires us to make these decisions based on existing research and theory, so having this information means we don't go in as blindly and we give our plants the best chance possible. Beyond understanding the ecology of a species, ex situ collections can be tremendously important for preserving species and their populations from extinctions in the wild. A really good example of this, as Aline was talking about, was actually a landslide, which occurred in 1995 at the Glentilk natural population. This reduced the natural polygonatum population down to just 2 shoots. So our team at RBGE obtained a license to go out and collect a piece of rhizome, which was then brought back to the nursery and used to bulk up a collection. These plants were then reintroduced to the provenance in October 2007, so restoring those numbers and possibly even saving that provenance from extinction. 30 years later, we're still benefiting from these ex situ collections today. That one piece of rhizome makes up quite a lot of the planting material we use, and there was also 3 other provenances collected in October 2005, which make up pretty much most of what we use on the project today. It doesn't end there. In the future, frequent monitoring will be the only way we can review our success rate and the techniques of our translocations. Through all of our plantings, we've planted across different soil types, habitats, exposures, aspects. It's all one big trial. And by monitoring for what conditions are successful for the polygonatum, we should be able to create detailed planting protocols for future translocation efforts. This way, we can ensure successes of our translocations, guide horticultural care of these plants, and direct further research and resources into these species. In doing so, we give these endangered Scottish plants the best possible chance of recovery. Thank you for listening.